Okay, well, thanks very much for the invitation to come and speak at this. Anyway, I'll start just by, if I can move the slide on, I would. There we are. It finally moved. So I'll better introduce where I'm from, the James Hutton Institute. Just a few little facts about it. We are apparently the biggest agri-environment independent research organisation in the UK. And that gives you an idea of the number of staff and students. And basically we do a lot of different things, and that shows the range of disciplines and who we try and serve in the work that we're doing in the science community and broader. One thing I just want to mention, though, because some of it's relevant to the talk, is we've got long-term research platforms, and as I was just mentioning, as we're starting some flagship initiatives and some investment in the place at the moment. So the long-term research platforms, we've got the Greaves House, which is a tillage platform, which has been running for a number of years, and some of my work's involved in that. I'm not going to speak about that today. The CSC, the Centre for Sustainable Cropping, I will actually be referring to later on, because that's a big rotation experiment that's been running for about 12 years now, where we basically compare 6 large field rotation where there's 2 management types, one of which is conventional and one of which is integrated. And I'll be referring to that later on. The investment that I was showing in my background slide is the International Barley Hub and the Advanced Plant Growth Centre, which is a big new initiative, infrastructure investment, which is really encouraging to see investment in that. And we hope that will really bring together the broader community in those areas. And we also have the International Land Use Study Centre, which is based in Aberdeen. Also just want to mention briefly, we have some farms, Balruddery, which is our arable farm, which is where a lot of the arable experimentation is done. The main Dundee site is Millfield, which is a farm as well, and that's where most of the horticultural work is done. And then we also have a 1,000-hectare upland estate called Glensoch, where we have a new initiative there called the Glensoch Climate Positive Farm. Anyway, so that's just a little bit of background on where I'm from. And of course, that was also where Johnny did his PhD. So on to what we're talking about here, and just a bit of very general context. The context for this work is really things like biodiversity decline and biodiversity and ecosystem function. And very simply, context is as you increase biological diversity, you get an increase in ecosystem function. And in the context of a crop, of course, monoculture to even a 2-component intercrop, mixed crop, you're getting potentially a large increase in function, ecosystem function occurring as a result of doing that. And we're really talking, again, just basic context is it's about niche differentiation. So below and above ground, better exploitation of resources in that sense, and also facilitation, the interaction between plants, resource sharing, increased nutrient availability, and pest and disease resistance. So that's the context of mixed cropping in a way. And very briefly, various features of mixed cropping. 2 or more species grown together. We often refer to them as plant teams, particularly for the EU project that we just finished, which I'll refer to in a minute. Diversity often equates to increased biomass, increased resource use efficiency. The complementarity of competition and facilitation leads to over yielding or a greater than 1 land equivalent ratio, which I think Martin might speak about later as well. And the real benefit there of better resource use, greater resilience, reduced inputs potentially from the efficiency, maybe reduced loss as well, increased biodiversity. So it looks fantastic, doesn't it? Why isn't all agriculture like that? So, and I think one of the major things which I want to talk about this, and this isn't all the reason, but there's a lot of inconsistency, a lot of things we don't understand about how to optimize mixed cropping, which results in very variable outcomes. and a lot of difficulty in actually predicting the outcomes. And I'm not sure I'm going to answer many of these things. I'm just going to throw some thoughts out as to what some of the reasons might be. And in particular, and I delve into that in a little bit more detail, I'm actually going to refer to a system which has a fairly narrow range of diversity, which is variety mixtures, which I've worked on quite a lot over the years, where really you've just got, you're in the same crop species and you've just got variety differences, so some quite narrow differences. Nevertheless, they produce the same features of mixed cropping. So mixed cropping, lots of other names for it there, which I won't bother, you can read them for yourself. Just a point to make, mixed cropping isn't all about the cash crop, the main crop. It can also be a catch crop, a cover crop, green manure, biomass crop. They're all appropriate for mixed cropping, for getting more out of the system, getting more complementarity between the components. Here's a slide stolen from Eric Just via Ecological. Basically shows a lot of nice pictures of some really nice looking intercrops. They look very tidy, and these are the ones which are probably highly successful. And again, a bit of something from the literature, basically showing how many of these systems here of intercropping do result in a positive land equivalent ratio, and all significant results there. But I would actually say, what about all the ones which are not reported? What about all the cases where things don't quite go according to plan? We tend not to produce so many papers describing those. And maybe the overall picture isn't as nice as we sometimes can present by bringing together the positive cases where things have worked. Now, we've just finished earlier this year a big project, an EU project called Diversify, and Martin was involved in this. I think even Johnny worked on it for a short while as well. And this was a really useful project, and it started from what do the farmers and the practitioners know about intercropping. So we had workshops with them to find out what they were doing, what worked, what didn't work, and things like this. We then actually had participatory research where the farmers actually grew particular crops and reported on them, and we fed that all in as well. Meantime, the people from the more science background were doing the more detailed experiments and maybe trying out some of the things which came out of these workshops. And the idea of the project was really to bring all these things together, try and work out what is working, what isn't working. Let's communicate all this, let's get the benefit of everyone's experience, and let's develop it in a positive direction where it will be more reliable. And let's see what the issues are that need working on in future. And over the right just shows you the various countries involved in this. But we came up with various resources, and some of these are available. I'll show you the website right at the end where you can access some of these outputs. Also, currently in Scotland, we have another project which does something similar in a way, because it's also involving farmers, and they are doing the research. They are doing trials on their farms with whatever the relevant crop is to them. We're trying to get them to do not just the mixture, but also the single crops alongside, so that we can actually make comparisons. And then we're actually taking samples from their crops and also looking at that in detail ourselves, so comparing directly, but also getting their data. So we're actively involved in that. We're trying to get this network of core and network farmers working together, talking together, and they're all finding it really beneficial. And it's very much we're learning different things because we're doing different types of work, but bringing it together and having the benefit of that. We're also trying to get this into people's thinking a lot broader than just the farming community. So we're going to get, when COVID permits, school children coming out into the field to actually measure some of the effects going on. We're doing things for classrooms, and we have a lot of different organizations involved communicating this. So it's about how do we do cropping? It's about the mindset that we've got at the moment, but maybe the mindset that we should have in terms of getting the best out of the ecology of the crop. and a much better system. One of the things to mention in this context is that in Scotland, as an example to go alongside the other project, is there are increasing numbers of local products from intercrops, short supply chains, niche products, where part of the value of the product is actually coming from the fact that it was grown in a certain way, it was grown locally, it involved local people, and it was processed through to a crop locally. And I think that, again, is a really encouraging part of the overall work of things going on, and absolutely essential that it's not just researchers trying to develop it and spread it out there, but it's coming both ways, it's working together. So on to something about actual intercrops and maybe some of the challenges, but some of the benefits as well. And I just want to illustrate this with a few examples. So dependence on agrochemicals, or in particular herbicides in this case. And these are data from that Centre for Sustainable Cropping that I was talking about earlier, one of our facilities, where we've got conventional management and integrated management. And here we've got wheat-bean combinations and pea-barley combinations. And what we're looking at here on this axis is the weed biomass. And if you just look at the ones that I've ringed there, you'll see that the mixture in the grey has got lower levels of weeds in most cases, but also you get a difference between the two farming systems. It doesn't come out the same in every farming system, and every farming system is different for different reasons. Here's another example from the literature. This was the one from the Remix project, I think, which was looking at disease incidence. And this just shows lower disease of farmer beans and cereals from mixtures compared to the expected at different nitrogen levels. So it's not unique to particular nitrogen levels in this case. Here's another one from our CSC, so conventional and integrated farming practice. And what we've got here is the number of aphids on the monoculture and the reduced number on the mixture. So again, it's working out well there, but the two systems here have different results again. So it's always different depending on where you're actually implementing it. Here's another example from the literature. Peas and barley grown in monoculture, peas and barley grown in intercropping. And this is the increased nitrogen fixation by a legume in the two systems. Not a lot of difference there, but improved soil nitrogen uptake by the cereals. Yes, it was increased in the intercrop, possibly because of more going to the cereal component in there as opposed to the legume component. Grain nitrogen actually is up sometimes in mixtures compared to monocultures where you've got a legume as well, as the example on the right there. Here's another example from the CSC, conventional and integrated farming again, and this is soil phosphorus. And again, in the conventional system, you'll see that the mixture here has high levels of soil phosphorus. In the integrated system, it made no difference. I've talked so far about barley, about wheat and peas and beans, but actually, of course, it's varieties. And these data here just illustrate how important varieties can be. This again was some work that we did at the Institute, where what we've got down this left side is the pea varieties, so Clara, Daytona, Ingrid, Sakura. On the right-hand side are 4 barley varieties. And I've highlighted the ones where you get more than 20% increase in yield over the expected in the monocultures, i.e. 1.2 equivalent ratio. And you'll see that they're all in this middle group. In other words, they all came from Daytona or Ingrid, whereas there are lower levels from the other 2 varieties. So it's quite a strong variety effect that you often get. It didn't seem to make any difference which barley variety you used. Here's another example, same color scheme here, but this was wheat and bean. And here you'll see it was actually the wheat variety made the difference. So Tybalt was delivering the more than 20% extra, Alderaan was not. So variety seems to have a strong effect in many of these systems, but probably for a number of different reasons. Here's another example, a different project. This was winter peas and winter barley and these crops in the plots in the background here. And it's a challenge to get winter peas to grow well in Scotland. And actually the cereal makes it successful because it stops it just collapsing. But you'll see down here, we've got commercial breeding lines of the peas. And here it was either done under direct drilling or under ploughing. And you'll see very different response. The peas basically don't do very well under direct drilling. They do much better under ploughing. But also some of the peas do comparatively well, even under direct drilling. And they do well under the plough system here. They probably are really almost out competing the barley in this particular case over here. So again, this is just a feature of different systems that we've chosen to highlight there, making a big impact on how the mixtures work. Now I want to go off into a little bit of detail of the mixtures, blends within barley situation as well, to try and illustrate some of the things which may be happening, and maybe you can offer an explanation as to what is actually happening. Now, this first slide is a really ancient slide I've been using for many years, but it illustrates the principles of cultivar mixtures very well. So here we've got two component mixtures, 3, 4, 5, 6, and this is the amount of reduction in disease. This is a winter barley, and the disease was ringsporium or scald. And you'll see that you get about 50% reduction with the complex mixtures, simple mixtures only down to about 20%. So that's the normal trend you get. The greater complexity, the more mixtures efficacy you get. In this case, for disease, it came through in yield as well. So this line here shows the control, so where there was still disease, and again, the complexity delivered more. But even where you took the disease out by using fungicide, you still got the same relationship. So there were still interactions between the plants apart from disease control as well. So that's the basic way, and I've probably got lots of data that I could show you that happening, where it works better in the complex mixtures than the simple one. And then winter barley is pretty reliable. Well, that's what I thought. And say these are results from my conventional system. And then I actually spoke to this guy called Greg Painter in Australia, and he did similar experiments. And this is just one that was reported. How many others were not reported? Where he did complex mixtures of winter barley and had no yield benefit. He didn't get a negative either. He just got no effect, no interaction really. Different climate, different varieties, different agronomy. Maybe the reason is tied up in those, but the point is, it's not translating to a different system. Why not? And what if we wanted something that translates to a, let's say, a low input system, organic system, a low density system? Maybe we want to use mixtures together with other crops in mixtures as in intercrop systems. How do we get these to reliably work? I think what I'm asking is, how do these different factors affect the competition facilitation sort of balance between components, and how do you choose them? So I want to consider what winter barley is, elite winter barley. How was it bred? It was bred under high nitrogen conditions in general, because that's the way we grow it in practice. It was bred under high density conditions, about 360 plus per meter square. Elite spring barley, maybe a bit lower because it's used for malting in particular, are the ones that we grow here, maybe a bit lower nitrogen, but high density again. So fairly intense selection conditions. Let's now compare this to what I'm going to term land races. So in the case of winter barley, these are old varieties bred at a time when they use much lower nitrogen. And also landrace or things like the Scots Bears, spring barley varieties, much lower nutrient conditions. They were selected or bred under different conditions. I don't know about this, but I suspect that they were probably all under lower density as well, so lower competition. So I use these in some trials. So I'll take you through these, hopefully not too fast so that you understand what I was doing here. But start off here, I've got Optic, which is an elite spring barley here. And then I've called them bears, which are actually 5 different Scots bears. So landrace type spring barleys. And I mix them in 50-50 combinations, or 90 of the Optic to 10 of the bear. No added nitrogen. And it gave, in this case, about 4% more yield than the mean of the monocultures grown separately. In this case, about 4% less than the mean of the monocultures. Change the variety from Optic to Irene. Same conditions, same nitrogen, no real interaction going on there. So really there's quite strong genotype effect coming in there. Do exactly the same under normal rates of nitrogen for barley. So now they've got the amount of nitrogen they've been bred for, or rather the elite element has been bred for. And suddenly we're getting good yield, positive interactions going on for them all, really, fairly similar. Now that had disease in it. What if you take the disease out? Will it be the same? So put fungicide in. What happens? Well, where you've got full rate of nitrogen, pretty much the same is happening. Where you haven't got, you've got probably insufficient nitrogen, so there's a strong competition going on for what little nitrogen there is, maybe. You've got much stronger interactions going on, it seems. I tried something else, which is a bit unusual. I also did the same trial under conditions of continuous barley growth. So the site had had continuous barley for the previous 10 or 15 years, which normally depresses barley yield. But what would that do to a mixture? I really didn't know what to expect. And basically, if you gave them enough nitrogen, it really didn't do anything to the mixture. Again, all of these, all these pretty much the same sort of response. But again, you're even getting negative responses when it comes to these here. Different circumstances, different interactions going on. What's interacting with what and why? Explain the second experiment now. I've extended what I did previously, but I've got mixtures of elite varieties. And these are all the, I think it's 5 or 6 different elite varieties, and all the 2-way combinations and all the 4-component combinations. And I grew these at, as in previous experiment, no added nitrogen, optimum added nitrogen. Also, I added half the rate of nitrogen and 50% more. I did this also at not just the 360 per square meter normal density of the previous experiment, but also at a low density. And look at, for example, here, high competition and low nitrogen, low resource, if you like here. And you get positive effect. Go to the other extreme, high nitrogen, low competition, and you get a negative response. That's just about a suggestion of a trend going on across them there. What about if you did this rather than the elite varieties, the bears instead? And this is what we've got. The opposite. Under high competition, low nitrogen, negative effect. Low competition, high nitrogen, positive effect. So has it got something to do with what these varieties are bred for? How they are bred? Are they bred to compete? What are they competing for? With each other or with the nutrients? I said that was the 2 and 4 component mixtures. This is just the 4 component mixtures. With the more complex mixtures, you get stronger effect generally. And you'll see that, for example, here, you get up to 12%, 10%. So it's exaggerated the effect a bit. Now, this is spring barley. Now I want to bring in winter barley. Winter barley is quite a different crop. Winter barley obviously goes through a long period of relatively high stress when it's not growing, then ripens sooner. So it's really quite different crop. It's barley, but it's still very different in the way it actually grows in practice, and probably therefore all the dynamics of how the components interact. So did the same thing here. Let's wipe out those. So we got just the comparison between the same 4 component mixtures. Again, in each cases, there are about 5 different varieties in all the possible 4 component mixtures. Again, we've got the elites, we've got the land races. Again, we've got exactly the same nitrogen range here, and we've got the same 2 different levels of competition. And if you just compare the elites, here you're getting positive, there you're getting negative. In this case, you get negative in the spring barleys, and in the high nitrogen, low competition, you get positive there. Very different crop. But then look down here at land races, and in winter barley, they're behaving in the same sort of way as the elites. I'm going to be asking you for explaining this to me afterwards. I mean, we've got competition difference between the 100 and 360. We've got more resource going in that direction as well. It must be something to do with how that then affects the competition facilitation between the components. And I mean, at the moment with winter barley mixtures, they seem to behave more like landrace spring barley, not like elite spring barley. Can we tell something about this? That's what I'm trying to work out. But it may be, once we translate, the same sorts of interactions are going to be happening at maybe a more exaggerated scale with intercrops with different species. So now on to just in the final bit, just move on to something else after hopefully our brains were working hard on that. And this is something else that came out of the Diversify project, because really this is about the challenges of intercrops, what I'm trying to talk about here. And we asked people who grow intercrops in practice what they perceive the challenges to be. And you don't need to really read all this, but they came up with a heck of a lot of challenges at these workshops that we did. And a lot of them came down to they really needed more information and advice, and hopefully we made some inroads towards that during the project. They needed to know there were a lot of agronomy issues, processing, economics. So it directed us towards those which were, some of these could be easily resolved, some of them couldn't, and some of them were maybe even perceived and maybe not the actual problem. But they needed solutions to actually improve delivery of mixed crops in practice. And I just want to focus on one area where quite a lot of work was done by the team at Stockbridge Technology Centre. Jen and Dave and Andrew produced an excellent report on this, where they looked at how precision agriculture technology might be able to help. I'm not going to go into the details of, because there's so many different options, but they basically classified the systems or solutions depending on whether they'd work for structured mixtures, where you've got the mixtures in particular formats in the field, or unstructured, where they're quite random. And they also assessed them whether they were already there, solutions accessible. Maybe they needed a lot of development and adaptation, or there were things which really were for the future, but had potential. And there were things like drilling barriers. How do you deal with multiple seed rates and different depths and different sowing times and seed sizes? And then in agronomy, it's weed control, disease control, competition, the sort of things maybe I've been talking about there. And this one kept on coming up, and an existing problem with a lot of them is things like seed separation afterwards. How do you actually deal with that in practice? In terms of unstructured plant teams, there were some solutions at all sorts of different levels for each of these various areas. In the structured plant teams, maybe there were more solutions because you knew where the different components of the crop were there. So that's almost solves one of the issues. So we identified quite a lot of potential, and we're trying to work with companies and maybe in the future develop solutions that could help there. But just returning to seed separation, that being a very basic one that keeps coming up again and again, people of course already try and do this. And this is a farmer we work for, Doug, and he has this lovely old seed cleaning machine, which he uses to sort out seed, and it works for if you've mixed wheat and beans because they're massively different in size. It's a very old setup that does the same sort of thing. But how do we do that more reliably for all sorts of crops? So what we did is put this to a load of mathematicians, and they came up with different thinking because, I mean, seeds vary in length, width, mass, density, color, all sorts of things. And so they started thinking about things like fluid dynamics and all sorts of things like this. And you can use pressure and image analysis and different physical shapes and things. And so really, if there's enough people doing this sort of work, and if the problem is big enough, there's probably a lot of solutions that we can come up with if there's the impetus to actually do that and develop a solution and then commercialize it. So that, again, one of the advantages of all this work, bringing together and bringing them up in profile, and the Remix project did the same sort of thing as well, is that it then creates a community which can have the pull to actually demand and develop all the technologies which again do implement mixed cropping much more practically. So that's me just about finished. I want to just leave you with this, which is the Diversify Legacy website, where if you want some more information on all things like the practical guides we developed, the decision aid that we developed in the work as well, and also all sorts of things for policymakers as well, to try and further this sort of work and the advantages of it in practice, then that's a good place to go. And there's many other things I could have spoken about in this, but I've hopefully tried to just illustrate some of the problems and maybe some of the ecological things going on underneath, which we maybe understand something of, but need some direction in understanding more and developing some principles and rules to develop these further and improve our choices and be able to actually develop more reliable intercrops. So finally, the funders, you can read them for yourselves. Enormously grateful to them all, especially the EU for some of the things that we talked about there, Scottish Government, bread and butter for some of us in Scotland, and many other funders there as well. And so just say thank you and be very happy to discuss any of the points I brought up.